So this little video is meant to help you prepare for our protein analysis lab, where you are going to be determining the concentration of protein in 2 different food samples that you are bringing to lab. And so I want to give you just a little bit of background about what exactly is a Bradford assay, what's the theory behind how this works in order to determine our protein concentration. Then I'll talk to you a little bit about what you should expect to see in your samples as you prepare them for the spectrophotometer. And then the last part of this video will discuss sort of what to do with all of the absorbance data that you're going to be collecting. So how do you construct the graph, the standard curve, and how do we use that standard curve to determine the protein concentration of your food samples. So the key assay that we are going to be doing during the protein analysis lab is something referred to as a Bradford assay. A Bradford is used to determine the protein concentration in an unknown sample that you have isolated. So maybe you've done an experiment and now you need to isolate proteins from cells that have been growing in tissue culture, from particular tissue from an animal, or something like that. And then you're going to turn around and use that protein that you've isolated in a different set of assays and experiments. Maybe you're going to run Western blots, maybe you're going to do some other functional assays. It's very important in those experiments that you are adding the same amount of protein across your different samples. Recall, when discussing experimental design, you really only want to change one variable at a time, and you want everything else to be constant. Since you're usually looking for other readouts, it's important that your protein concentration is consistent across samples. And so the Bradford is helpful because it allows us to know what concentration my isolated sample is, and therefore how much do I have to load in every sample in order to have a consistent concentration. So the basis of this assay is to use something called the Bradford reagent, and the key component of the Bradford reagent is a dye that is referred to as Assay. Assay will specifically bind to the amino acids found in proteins. And in particular, Assay will target positively charged functional groups on amino acids. So it doesn't just bind anywhere, but it binds to all those positively charged R groups. When the dye binds to our protein, it forms a complex. And this complex will change color. So the Bradford reagent on its own in the bottle is a kind of drab brown color. But when it forms a complex with our protein, it changes color to blue. That color change, that shift from brown to blue, alters how much light is absorbed by our particular sample. And we can measure changes in light absorbance using a spectrophotometer. And so in particular, since we are looking at absorbance by proteins to determine protein concentration, usually we read that absorbance at 595 nanometers. And so in your assay this week, that's what your spectrophotometer is going to be set to read at. The key with the Bradford assay is that the more protein you have present in your sample, the more blue your solution is going to turn. So if by the time you're all said and done with your experiment and you look at the color of your 2 food samples and they're sort of blue, but they still kind of look a little bit brown, that's going to immediately tell you you probably don't have very much protein in your sample. It's a lower concentration. If, however, when you look at your food sample after mixing with Bradford, it is a deep, brilliant, intense blue, automatically, just by looking, you will recognize that you have a lot of protein in your sample. Now, it's going to be important when you run this assay that you add Bradford not only to your 2 food samples, but the Bradford reagent also has to get added to all of your standards that you are going to use to generate a standard curve. Now, typically your laboratory instructor will have the standards prepared for you and you'll just have to spec them, but do know that it is very important that Bradford has been added to those samples as well so that we can actually measure their absorbance. So once you have incubated all of your samples with our Bradford reagent, you will end up with a string of cuvettes that looks like this. Now again, I believe your lab instructors will be creating your standard curve. And to create your standards, we are using a protein source called bovine serum albumin, or BSA. This is a very commonly used protein source in molecular biology because it is very stable and it doesn't interfere with most of the chemical reactions that we are usually trying to investigate, so it's relatively inert. What you will notice is that we are using a variety of different concentrations of BSA in order to create our standard curve. So at the low end, we are using a concentration of 0.125 milligrams per mil all the way up to 2 milligrams per mil. Okay? So at 2 milligrams per mil, this is our most concentrated in terms of amount of protein, or you could also refer to it as the least dilute sample. Notice that really intense blue color as there is more protein to interact with the chemosity. At the lower end of our Bradford standard curves is the 0.25 milligrams per mil. Notice that this color is much closer to the original brown of our Bradford reagent. This is our least concentrated protein standard, has the least amount of protein in it to react with the chemosity, and is therefore our most dilute standard. Notice as you move from that sample up to our 2 milligrams per mil, you get a nice shift in color from that brown to a blue. When you go to the spectrophotometer, you will also need to have a blank, okay? This is sort of your control, and you're going to use it to blank the machine and set the absorbance reading to 0 to start your assay. Your blank is typically just going to be your diluent for your samples, and in our case we're using phosphate buffered saline, or PBS, and then you add the Bradford reagent to it. Since there is 0 protein in PBS, you're not going to get a color change, and so your blank should look brown. If it doesn't, you've got contamination and you want to reset up your blank. Then you will also have 2 of your protein samples, right? 1 from each of your food sources. You may even be able to take your food samples and just compare them color-wise to these BSA standards that have been generated, and just by looking at the color you can sort of predict where you think the concentration of protein in your sample is going to fall. So this is what your samples should look like that you will be taking to the spectrophotometer. And so this is an image of the spectrophotometer that we will be using in this course. So these are Bio-Rad Smart Spec Plus, and this is what we will be reading all of your samples in. Again, for protein concentrations, we want to read our light absorbance at 595 nanometers. And again, your laboratory instructors will help make sure that your spec is set up properly. The first thing you will do is start by blanking the machine, and that means the first thing that you're going to want to read is your blank cuvette. This basically sets the absorbance reading of the machine to 0 so that you can then read all of your actual samples. After blanking the machine, you're going to want to begin by reading all of the standards that were created with the BSA. So you are going to read the standards one at a time, and typically we start with our least concentrated, right? So tube 1 was our least concentrated at 0.125 milligrams per mil. Start with that cuvette first, and then work your way up to that most concentrated 2 milligram per mil sample. After you have read all of your BSA standards, then you can go ahead and spec each of your 2 food samples. Do not blank the machine in between reading your standards and your samples. As soon as you have read your last BSA standard, you go right into reading your food samples. You do not need to blank the machine again. Also very important, when you are doing this, you are placing the entire cuvette into the machine so that the light can pass through your sample and the absorbance be read. Please do not pour your samples into the machine, as this will destroy it. Actually place the entire cuvette into the machine. I only say this because I have watched it happen and I don't want it to happen to you. Okay? So again, blank your machine, run all your standards first, least concentrated to most concentrated, then read your 2 food samples. Now at this point, lab is done. You've spec'd all of your samples and you've got absorbance data. What now? Come back to this portion of the video after lab and we'll talk about how to use your data to generate a standard curve, how to create that curve using Excel, and then how to use your standard curve to determine the protein concentration of your 2 food samples. So now that you have your absorbance data, we're going to want to use the data from your BSA standards to generate a standard curve. This curve will then allow you to figure out the concentration of protein in your food samples. So since this standard curve graph is going to be part of your first lab report, we're going to ask that you create it in Excel. Now maybe you're an old pro at using Excel. If that's the case, you can ignore the rest of this video and go ahead and make your graph. If maybe you are less familiar with Excel, I'm going to walk you through how to create the graph in that program. So you want to go ahead and open up Excel, and in your spreadsheet you're going to want to create 2 columns. The first column will be the concentration of your BSA standards. So remember, these were known concentrations that we gave you from 0.125 to 2 milligrams per mil. The second column you're going to want to create has your absorbance values that you got from the spectrophotometer. Now mine is fictitious data that I just created in order to generate a curve to show you how this works. So your values might be completely different, and that's okay. Now in creating a curve, your gut reaction is probably going to be to include all of your samples, including your blank. But realistically, when we create standard curves, we do not include the blank on our graph. And so I'm actually going to go ahead and delete the values for my blank. So I really only have values for my actual protein standards. In order to generate your graph, what you have to do is first highlight both columns of data. So you're just going to put your cursor here, drag it over and drag it down and highlight both of these columns. Then you're going to go up to Insert, click on that, come over here to the chart area, and you're going to want to click on this one right here that looks like a bunch of dots. So we want to generate basically a scatter plot for our particular data set. So you're going to go ahead and pick scatter plot. And that will generate a graph that looks like this. Now this graph is going to need to be manipulated in order to make it look like the type of graph we would see in a peer-reviewed journal article. So what you're going to want to do to finalize your graph is to first go ahead and delete the chart title and all of the grid lines. So to do that, you can, in Excel, just click on chart title. It'll highlight it here. It'll create a box with little blue dots, and then just delete it. You also then need to highlight your vertical grid lines and delete them, and click on your horizontal grid lines and delete all of those, because we don't see those in graphs that appear in journal articles. So that will get you a graph that looks something like this. Now what we do need to include are labels for our X and our Y axes. In order to add these labels, you can go up to the chart tool. So you have to have your chart selected. So notice I have my chart selected. Go up to chart tools, click on design, go all the way over to the left and say add chart element. That will give you a drop-down menu where you're going to want to choose axis titles, and you're going to need to add both a horizontal and vertical axes. There is a shortcut to get to this option, and that is to click on this little green plus sign here, and it will give you the option to add axes titles. Keep in mind that your x-axis is your independent variable. It is what you, the experimenter, are manipulating. In this case, it is the concentration of the BSA in each of our standard samples, and that concentration is in milligrams per mil. Your y-axis represents your dependent variable, what you are measuring in your experiment. In our protein analysis lab, you are measuring the absorbance of your samples at 595 nanometers. And so those are the labels you're going to want to add to your axes. And so you can see in my chart here, now I've got my x-axis label and my y-axis labels hidden, but you can see it peeking out here. Now that you've got your labels, you want to add your best fit line. And now Excel can do this for you instead of trying to do it by hand. To add your best fit line, again, you're going to go to Chart Tools, Design, Add Chart Element. Now you'll scroll all the way down to Trend Line, and you'll want to add a linear trend line. So now you can see the dotted best fit line on our graph. Please note that best fit lines are not connect the dots. The idea with a best fit line is that you take all of your data points and they are evenly represented so that half are on one side of my best fit line and half are on the other. So for some of my plot points, like this one, you can see the line goes right down the middle. But it's a little bit off for some of the other, and in fact this data point here is fully on one side of the best fit line. Okay, now again, Excel will do this nice and neatly for you, but that's something you would want to keep in mind if you're ever graphing by hand. Once you've got your best fit line, in journal articles, we typically also represent the slope equation for a best fit line and the r-squared value, which tells us something about the quality of our best fit line and how much do we trust that data set. You can have Excel add these equations directly to your chart. So what you would do is, again, go to Chart Tools, Design, Add Chart Element. If you scroll all the way to the bottom to Trend Line, there's an option that says More Options. If you click that, you should get a display on your screen that looks like this gray box off to the side, Trend Line Options. Scroll all the way down to the bottom. You're going to want to check Display Equation on Chart and check Display R-squared value on Chart. And here we can see those equations quite nicely displayed on our graph. And so at this point, you have a graph. just like it would appear in a peer-reviewed journal article. This graph is going to end up in the results section of your first lab report. And so now you can very easily select this chart and copy and paste it, in your case, into a Word document. So I've copied and pasted my chart into my PowerPoint so I can continue to talk to you about how we're going to use our standard curve. Please remember that when you are embedding this graph into your lab report that you need to include a figure legend. So again, we don't use chart titles, we use figure legends. Since this is your only figure, it will be figure 1, and after figure 1 you need to have a descriptive sentence or two that's telling us exactly what is being displayed or depicted in your graph. The reason you made this standard curve in the first place was to determine the concentration of protein in the 2 food samples that you brought to lab, right? And so you know the absorbance value for your 2 samples. It's what you measured on the spectrophotometer. Now we're going to use our standard curve to figure out their approximate protein concentration. It's important to use absorbance values of your food samples that fall between your lowest and highest protein standards. So I've kind of highlighted those for you here with my pink dotted line. Ideally, in order to be able to interpolate your protein concentrations, your absorbance values of those food samples will fall in between those 2 data points, somewhere on your best fit line. If your absorbance is higher than your highest BSA reading, then you can only extrapolate, and it's not quite as accurate as using data points that fall on the best fit line. So hopefully your dilutions worked out and you have absorbance values within your standard curve. You're going to want to use the graph to interpolate the concentration of both of your food samples. And so I have my first food sample in green here. And so again, to interpolate, you're going to find your absorbance reading on the y-axis. You're going to go in a straight line over until you hit your best fit line, and then you will drop straight down to approximate your your protein concentration. Do the same thing for your second food sample. So I have that indicated here in purple. Find the absorbance reading that you measured of your food sample, go straight across to your best fit line, and then drop down to approximate your protein concentration. Now, that is not necessarily the actual concentration of your protein in your food sample. Chances are, depending on what food you picked, your sample was diluted. You have to account for that dilution factor when calculating the concentration of your protein. To do that, you merely take whatever value you read off of the x-axis. So for sample 1, I said it was approximately 0.5 micrograms per mil. To find the actual concentration in your sample, you need to multiply by your dilution factor. So let's suppose you diluted your sample 1 to 10. That means you would multiply 0.5 by 10. If you had done a 1 to 50 dilution, you would need to multiply 0.5 by 50. You'll need to do the same thing for sample 2 if it was also diluted. Depending, you may not have had to dilute your sample. If that is the case, then you do not need to dilute your dilution. Let me start over. If you did not have to dilute your sample, you will not need to multiply by a dilution factor. Okay? So you only use the DF if you actually had to dilute your samples. And so now you've created a standard curve that can be embedded in your lab report. You have used that standard curve to interpolate the concentration of your 2 food samples. And now you can start to draw conclusions on your data. You can think about, how does the protein concentration you determined align with what's reported on the nutritional label of your food sample, and how does the 2 food samples compare to each other? So in my fictitious data set here, my sample 2 clearly has a higher protein concentration than whatever my food sample number 1 is. And that will be the basis of the results and discussion section of your first lab report. So hopefully you found this video helpful, and don't be afraid to reach out to your friendly lab instructors if you still have additional questions.